Hello everyone! This second video about the central dogma used in molecular biology will now concentrate on the process of transcription. As a recap, DNA makes RNA and RNA makes the protein. We have learned in the previous video that the DNA contains the biological information that is encoded in the nucleotide sequence, and DNA is able to self-replicate. The essential link between the biological information and how this information is projected or expressed in a living organism is now therefore made possible by the gene expression pathway. We must now turn our attention to this pathway and examine exactly how DNA is transcribed into an RNA and how RNA is translated into a protein. The gene expression pathway is the link between the DNA information and the characteristics from protein translation. In its simplest form, the gene expression pathway is looked on as a two-step process. The first step is the transcription, which results in the synthesis of mRNA and non-coding RNAs. The second step, which is translation, uses the mRNA to direct the synthesis of the proteins. There may be variations in the gene expression pathways found in different types of organisms. In some types, the pathway is much less complicated than the others. The prokaryotes, eukaryotes, and the archaea are the three main types of living organisms, with each group displaying its own distinctive genetic features. Of the three groups, the archaea are the far least well studied. And at present, the gene expression pathway operating in these organisms is not understood in detail. So, we must focus our attention on the different gene expression pathways in the bacteria, or the prokaryotes, and the eukaryotes. Transcription is the process where the gene's DNA sequence is copied or transcribed to make an RNA molecule. Therefore, the name transcription. The end product of transcription is the mRNA, also called as the RNA transcript. This serves as the molecular copy, which is mobile because it needs to be transferred out of the nucleus of the original DNA sequence. In eukaryotes, transcription happens in the nucleus. No nucleus is found in a prokaryote or a bacteria. As just mentioned, the transcription process involves copying a gene's DNA sequence. A gene is now defined as a segment of a DNA. There are two broad steps in the production of RNA. First, we have the pre-mRNA production and the mRNA production. For the pre-mRNA production, we involve three more steps, which is initiation, elongation, and termination. mRNA is produced after the pre-mRNA and is done through the process of RNA processing. M stands for messenger, so mRNA means messenger RNA. The first phase of transcription is initiation. And this is considered as one of the most important phases because it is a key control point in the gene expression pathway. Initiation starts when an enzyme or the RNA polymerase attaches directly or via accessory proteins to the promoter or the core promoter sequences. So this is the DNA, the promoter, and the oval is the RNA polymerase, and then creates an open promoter complex from a closed promoter complex. It does this by breaking a limited number of base pairs. True completion of the initiation stage is accomplished when the RNA polymerase has made a stable attachment to the DNA and has begun a full synthesis of a transcript. For short, initiation is the phase when the RNA polymerase attaches itself to the DNA through the promoter or core promoter region. After this attachment, it will open up or split the DNA in two. Involved in the initiation process is the promoter. The promoter is a sequence that is located in the DNA and it facilitates the transcription of a particular gene. The promoter or the promoter region is a sequence of base pairs located at the beginning or upstream of the gene or genes to be transcribed. This promoter region guides the RNA polymerase where to attach and where to start. So this signals the start for the enzyme RNA polymerase. This is an enzyme which is responsible in the transcription process. This enzyme transcribes DNA into RNA and is called as the DNA-dependent RNA polymerase, which we can always shorten to the term as RNA polymerase. There are also RNA-dependent RNA polymerases involved in the replication of some viral RNAs. The RNA polymerase are the central component in the transcription process. The main player in the transcription process is the enzyme RNA polymerase. Its main function is to copy the information from the DNA to the RNA. Like the enzyme in DNA replication, it also builds from the 5' to the 3' end. This is the RNA being produced by the RNA polymerase building from 5' to 3'. That means it needs to attach to the 3' end of the DNA template. Unlike the DNA replication though, the RNA polymerase only uses one strand, and we know this strand as the template strand. Now, this strand runs from the 3' going to the 5' direction. Another name for the template strand is the antisense. The other strand of the DNA, known as the non-template or the sense strand or the coding strand, is not used in the transcription process. This strand is not duplicated. Unlike in replication, the RNA polymerase zips back the DNA that is done being transcribed. As we can see here, the RNA polymerase moves forward. It opens up the strands ahead and contains the transcription process inside the open system and leaves the DNA strand zipped back. The second phase in transcription is elongation. This phase is the process of copying the bases on the DNA template strand to form a new RNA copy. The RNA polymerase adds nucleotides according to the base pair complementary rule to create the RNA copy. But one slight difference is that in RNA transcription, the RNA molecule contains uracil instead of thymine. So no thymine may be seen in the base pair sequence of the RNA growing end. The third phase in the transcription process is termination, which involves the stop signal or the terminator region. This, for example, is the stop signal or the terminator region. Now, once the RNA polymerase reads the terminator region, the newly formed RNA is released, the RNA polymerase dissociates from the DNA, and the DNA reverts back to its original structure. The RNA produced after termination is now called as the pre-mRNA or the pre-messenger RNA. This RNA contains the gene codes from the DNA that it transcribed. But this pre-RNA is not yet ready for the next step, which is translation. After termination is the RNA processing. For prokaryotes, however, the mRNA is already ready for translation. Therefore, RNA processing is not required. For eukaryotes, on the other hand, the pre-mRNA produced after termination needs to be processed before translation. So if this is our original template DNA, our RNA transcript has a copy of the gene that we need. But this pre-mRNA is not yet ready for translation. We have to follow 3 steps for the processing. First is the addition of the cap and the tail to the ends of the pre-mRNA molecule. These caps and tails are short amino acid sequences that will protect the pre-mRNA molecule because these are liable to attacks from many enzymes. The cap or the 5' cap is a modified guanine, while the 3' tail is made of a bunch of adenine. The next step is the removal of introns. Introns, as we can see here, so this is the whole gene. The introns are the green parts. That means these introns are considered as junk sequences. They do not contain any codes or coding that would carry information to build proteins. In short, these are non-coding sections, so they have to be removed before translation. All we need are the exons. The exons are the ones that code for proteins. And this is done by the enzyme spliceosome. Spliceosomes remove the introns so that only the exons will remain in the final mRNA product. This removal of introns is called RNA splicing. And lastly, the last step for the RNA processing is to join the RNA together. And this now is your mature mRNA. In bacteria, the target sequence for RNA polymerase attachment is called the promoter. A promoter is a small nucleotide sequence that is recognized by the bacterial RNA polymerase as a point at which it should begin transcription. Promoters just occur upstream or before or at the beginning of the genes to be transcribed. The E. coli promoter is made up of 2 distinct components, the -35 region and the -10 region. The nucleotide at this point is labeled as +1 and is anywhere between 20 to 600 nucleotides upstream of the start of the coding region of the gene. The -10 region is also known as the -10 box or the Pribnow box after the scientist who first discovered it. The -35 region, or the -35 sequence, is recognized and bound by the sigma factor. The sequence of the -35 box is described as 5' TTGACA 3', and the -10 box is described as a sequence of 5' TATAAT 3'. The most efficient promoters are called strong promoters, and they can direct 1,000 times more as many productive initiations as the weakest promoters. For the RNA polymerase in bacteria, the E. coli RNA polymerase is a large protein made up of several different polypeptide units. Each E. coli cell contains 7,000 RNA polymerase molecules, and between 2,000 to 5,000 of these are actively involved in a transcription at any time. All the E. coli genes are transcribed by the same RNA polymerase enzyme. Elongation is RNA synthesis, and the elongation in bacteria begins when the sigma subunit, the green part here, dissociates from the enzyme. This allows the core enzyme to synthesize RNA complementary to the DNA template in a 5' to 3' direction at approximately 40 nucleotides per second. As the elongation proceeds, the DNA is continuously unwinded ahead of the core enzyme and rewound behind it. The RNA polymerase covers about This is the RNA polymerase. It covers about 30 to 40 base pairs of the template DNA. This includes the transcription bubble. The transcription bubble holds around 12 to 14 base pairs, and inside this bubble, it also holds together the transcribed RNA and the DNA together of about 8 RNA and DNA base pairs. The polymerase does not synthesize the transcript at a constant rate. Instead, the synthesis is discontinuous with periods of rapid elongation and interspersed with brief pauses. The pause rarely lasts longer than 6 milliseconds and might be accompanied by the reverse movement of the polymerase, which is backtracking along the template. The longest bacterial genes are only a few kilobytes in length and can be transcribed in a matter of minutes by the bacterial RNA polymerase, which has a polymerization rate of several hundreds of nucleotides per minute. Once a gene is transcribed, the bacterial polymerase must dissociate from the DNA template and liberate the newly made RNA. This is referred to as the termination of transcription. Bacteria use two distinct strategies, but a common feature of both is that the termination site is marked by an inverted repeat in the DNA sequence. An inverted repeat is a segment of the DNA in which the sequence is followed by its reverse complement. This will form the hairpin loop in the RNA transcript. These are called intrinsic terminators. The second type of bacterial termination signal is RAW-dependent. Termination requires the activity of a protein called RAW, which attaches to the transcript and moves along the RNA towards the polymerase. RAW is a helicase, which means that it can actively break the base pairs, in this case between the DNA and the transcript, resulting in the termination of transcription. Prokaryotes and eukaryotes perform fundamentally the same process of transcription with a few significant differences. In prokaryotes, we place much greater emphasis on the start of the process. This preliminary step is more complicated in eukaryotes compared to bacteria, largely because the eukaryotes have more sophisticated mechanisms for controlling the expression of individual genes, and because many of these control processes operate by regulating the assembly of this transcription initiation complex. Eukaryotes use three different types of polymerases. We have RNA polymerase 1, 2, and 3. Although the three eukaryotic RNA polymerases have related structures, their functions are quite distinct. Each works on a different set of genes with no interchangeability. Most research has been directed towards the RNA polymerase 2, as this is the one that transcribes the genes that code for proteins. Therefore, this synthesizes the mRNA. But this is not the only role, because RNA polymerase 2 also transcribes genes specifying the small nuclear RNA and microRNA, which are two types of non-coding RNA. The other two types of eukaryotic RNA polymerases, 1 and 3, synthesize the non-coding RNAs only. In eukaryotes, the term promoter is used to describe all sequences that are important in the initiation of transcription of a gene. They include the core promoter. The core promoter is the site at which the initiation complex containing the RNA polymerase is assembled. Each of the three RNA polymerases recognizes a different type of promoter sequence. The promoters for RNA polymerase 2 are the most complicated, some stretching for several kb upstream of the transcription start site. The promoters for RNA polymerase 1 and 3 are not so complicated. In eukaryotes, there is an additional level of complexity. This is the protein coding gene that have the alternative promoters. Now, even though they are usually referred to as the alternative promoters, these are, more correctly, multiple promoters, as more than one may be active at a particular time. The TATA box, as you can see here, is a promoter for the RNA polymerase II, is the counterpart of the bacterial Pribnow box promoter. An important difference between the initiation of transcription of E. coli and the eukaryotes is that the eukaryotic polymerases do not directly recognize their core promoter sequence. For genes transcribed by RNA polymerase II, the initial contact is made by the protein called transcription factor 2D or TF2D. This is another multi-subunit complex made up of the TATA binding protein, or TBP, and at least 12 other TBP associate factors, or TAF. The TAFs, as seen here, and these guys over here, will assist in attachment of the TBP to the TATA box. The next step results in the attachment of the RNA polymerase. This RNA polymerase, they will now attach to the DNA. This involves two more transcription factors. It is 2B and 2F. 2B attaches to the DNA-bound TBP and forms the structure that the RNA polymerase II will recognize, while the TF will bind to the RNA polymerase and prevents it from attaching to the DNA at the wrong place. And then finally, we have two more factors, 2E and 2H. These are recruited to complete the initiation complex. 2H is very important because it is a helicase. This is a type of enzyme that is able to break the base pairs that hold the two strands of the double helix together. This activity is needed to convert the closed promoter into an open form. The fundamental aspects of transcript elongation are the same in bacteria and eukaryotes. Once successful initiation has been achieved, the RNA polymerase begins to synthesize the transcript. The RNA transcript is built up in a step-by-step fashion of 5' to 3' end, connecting to the 3' end of the DNA template. In contrast, RNA polymerase II can take up to hours just to transcribe a single gene, even though it can work to up to 2,000 nucleotides per minute. The reason for this is that there are presence of multiple introns in many eukaryotic genes, meaning that there should be a considerable length of DNA that must be copied. The three eukaryotic RNA polymerases each use different mechanisms for transcript termination. With RNA polymerase I, termination involves a DNA binding protein that attaches to the DNA at a recognition sequence located 12 to 20 base pairs downstream at the point of transcription termination. RNA polymerase II termination is accompanied by the addition of a series of adenosine nucleotides called the poly-A tail to the 3' end of the mRNA. Less is known about the RNA polymerase III termination. And that concludes this video about the RNA transcription in relation to the central dogma used in molecular biology. Thank you very much for watching.